We were interested in how the gut adapts to the availability of different nutrients. Now, we can think about animal diets as ranging in diversity. On one end of the spectrum, we have specialists like pandas that subsist on a restricted set of foods for their nutrition. And on the other end of this spectrum, we have generalists like ourselves, which can, of course, derive nutrition from a variety of sources throughout our lives. Now, these specialists have evolved fixed adaptations in the organization of their GI tract that allow them to efficiently extract nutrients from their restricted diets. But for generalists, we hypothesize that the gut must dynamically adapt to the availability of different nutrients that they consume. So to study this question, we designed two diets that differed only in the ratio of protein to carbohydrates, and we fed these to mice for 5 days. And after this time, we looked at the expression of enzymes and transporters involved in carbohydrate digestion in their small intestine epithelial cells, and we called this the carbohydrate transcriptional program. And what we found was that this carbohydrate transcriptional program was highly upregulated in the mice that were fed the high carb diet, indicating that this program is induced on demand in response to nutrient availability. We next thought about how this regulation might be occurring at the cellular level. And we considered that perhaps there might be changes in the frequency of epithelial cell subtypes that correspond to the breakdown of different nutrients. And so to address this question, we performed single-cell RNA sequencing of small intestine epithelial cells isolated from mice fed these two different diets. And indeed, we saw that when we compared mice fed the high carb versus high protein diet, we saw changes in the frequency of epithelial cell subtypes, indicating that the on-demand induction of this carbohydrate transcriptional program involves cellular remodeling of the intestinal epithelium. So in thinking further about the regulation of this program, we began to consider what other types of cells might be involved. Now, the small intestine contains the greatest number of immune cells of any tissue in the body. And previous studies over the past several years had demonstrated that epithelial lymphocyte circuits are critical for the intestine to defend itself against pathogens. And so we wondered whether these epithelial lymphocyte circuits might also be involved in the intestinal response to nutrient sensing. And so we iteratively tested the role of different lymphocytes, T cells, B cells, and innate lymphoid cells, and then alpha beta versus gamma delta T cells. And what we found was surprisingly that gamma delta T cells are required for the on-demand induction of this carbohydrate transcriptional program. And we went on to show that diet has a remarkable effect on gamma delta T cells in terms of their tissue localization and their transcriptome. And finally, when we investigated the mechanism by which gamma delta T cells regulate this program, we found that it occurs through an interaction with another type of immune cell called type 3 innate lymphoid cells. So during high protein diet, type 3 innate lymphoid cells, or ILC3s, upregulate IL-22. And IL-22 is a negative regulator of the carbohydrate transcriptional program. But during high carb diet, gamma delta T cells inhibit the production of IL-22 by innate lymphoid cells, thereby disinhibiting the expression of the carbohydrate transcriptional program. And so through this mechanism, we identified a novel role for gamma delta T cells in the intestinal response to nutrient sensing. But the most exciting part of this study for me was that we discovered that the immune system is playing a role in nutrition. So in my mind, the gut has two main functions: to take in nutrients and to keep out pathogens through host defense. And we understood that the immune system was playing a role in host defense, but what we didn't know was that the immune system also plays a role in nutrition. So the gut needs to adjust the balance between these two different functions, host defense and nutrient uptake, in response to pathogens and nutrients in the environment. But how is this balance being adjusted? I think the fact that we discovered that immune cells are playing a role in nutrition indicates that these epithelial lymphocyte circuits mediate the adjustment between host defense and nutrition at the tissue level. And I think the other surprising finding from this study was that we found that gamma delta T cells were doing this. Because although gamma delta T cells are highly abundant in the gut and other mucosal surfaces, we know very little about what they're doing during homeostasis. And so not only did we discover that the immune system is playing a role in nutrition, but we also discovered a new aspect of gamma delta T cell biology. So what are the limitations of this study? I think number one is that we worked in mice, and so it will be important to see whether our findings apply to other animals like humans and others. And then I think another limitation of the study is that we use these highly purified controlled diets. And of course, we did that because the question that we were interested in was the role of specific macronutrients in intestinal function. But of course, the diets that we consume and that animals consume in the wild are much more complex than the ones we used in this study. So I think in future studies, it'll be really important to look at diets with greater nutritional complexity and how those diets impact the immune system.